Hello friends, welcome to BMH Learning. This lecture is regarding gel electrophoresis. Before you get into this topic, you must have two purposes. Purpose number one, to understand the principle of gel electrophoresis. Purpose number two, to become familiar with the part of electrophoresis setup. Let's begin our discussion with the term electrophoresis. Electrophoresis is a separation technique that is based on the movement of charged particles in an electric field. The meaning of the word phoresis is being carried away. So the literal meaning of the term electrophoresis is to get carried away with electricity. Now let's know the principle of electrophoresis. It says any charged molecule or ion migrates when placed in an electric field. Now the migration rate of the particle depends upon the net charge of the particle, size of the particle, shape of the particle, and also the applied electric current. The principle can be represented in the form of following equation. It implies the velocity of the particle v is equal to the product of E and Q, which is nothing but the electric field applied and charge on the particle. The value which we get from the multiplication of E and Q is divided by the frictional coefficient. As we know, this lecture is about gel electrophoresis. So what is the meaning of gel in gel electrophoresis? The gel here acts as a spotting medium, like we discussed paper as spotting medium in paper electrophoresis. So the spotting medium is a material through which or on which a molecule or particle will move. Now, depending upon the type of biomolecules we want to separate, several gels can be chosen which are formed of different materials. So this gives rise to various forms of gel electrophoresis. First is agarose gel electrophoresis. Then we have starch gel electrophoresis. Next variant is polyacrylamide gel electrophoresis. We will discuss about each category in detail in separate videos. Now the important concept here is how the separation of the particles occur. So we will consider two important aspects. First is electric charge and the second one molecular size. Let's first talk about how particles are separated on the basis of charge. So many molecules like DNA, RNA and amino acids have naturally occurring negative and positive charges on them. Now the sum of these charges determine the overall charge. So let's suppose we have mixture of charged particles. Some of these are positively charged and the rest of them are negatively charged. Molecules with a negative charge will be attracted to the positively charged node, and molecules with a positive charge will migrate to the positively charged node. So the separation of molecules on charge basis is done. But if you see clearly, both separated charged molecules have different sized particles. Some of the molecules are bigger in size, some of them are smaller, and many of the molecules are of intermediate size. What next? Now they would be separated on the basis of size. So the particles are gonna move through gel. If that is the case, the gel should be porous, as you can see in the diagram. So what's happening is that the gel is formed of microscopic particles. These microscopic particles attach to one another, which forms microscopic tunnels. Now these tunnels act as molecular sieves through which the molecule will travel. Suppose we place a mixture of negatively charged molecules like DNA. The initial thing you are going to notice is that the larger molecules are the one which would be trapped in the net-like structure of the gel first. This would be followed by the separation of little smaller molecules than the previous large molecules. And finally, very small molecules will be separated, which still have the size smaller than the diameter of the molecular tunnel. This is how the separation of the molecules is carried out using gel electrophoresis. In the next video, we'll talk about agar gel electrophoresis. This was all. Thanks for watching.